Hello everyone, and thank you for being here today. My name is Bana Al Khairat, and I'm a master student in the Netherlands. In this presentation, I'll be discussing the high order adaptive dynamical system modeling for the role of the environmental factor neurotoxic pesticide paraquat on the epigenetics of neurodegeneration of N27 dopaminergic cells. This work is a collaboration with Jan Treur from the Department of Computer Sciences, Social AI Group at the Vrije Universiteit in Amsterdam. So before diving into the details, let me give you an overview of what we'll cover today. We'll start with some background on the topic, followed by the design of the high-order adaptive model. Next, we'll explore its functions. I'll give you a review on the simulation experiment, and then we'll conclude with a discussion on our model. To get started, I'll provide you with an introduction to the key concepts and the motivation behind the study. So let's start with a quick overview on Parkinson's disease. Also known as PD. It's the second most common neurodegenerative disorder, primarily characterized by a progressive loss of dopaminergic neurons in the substantia nigra pars compacta. To put it into perspective, the prevalence of PD is expected to exceed 12 million cases by 2040. Now let's talk about the role of epigenetics in PD. So epigenetic modifications are changes that can influence the gene expression without actually altering the DNA sequence. These changes are often shaped by environmental factors, for example, like histone modification, like acetylation, which we'll discuss in this model, that can regulate gene expression in response to external stimuli. So in our case for PD, disruption in these processes are linked to neurodegeneration and is associated with cell survival. And one key environmental factor linked to PD is paraquat, which is a neurotoxic herbicide. Previous research has shown that paraquat exposure contributes to dopaminergic neuronal degeneration through several pathways. The most importantly for us in this case, that it has been associated with epigenetic changes, particularly the hyperacetylation of histone 3, which has been linked to cell apoptosis in dopaminergic neurons. So to tackle these challenges, our approach involves modeling the epigenetic changes associated with PD using a high-order adaptive network. So this model uses multi-level self-modeling principles to explore the dynamic interactions between environmental stimuli like toxins and adaptive epigenetic mechanisms. So the goal is to better understand how these interactions influence gene expression and cellular behavior. So in conclusion, with the rise in PD cases and health risks by environmental factors like paraquat exposure, it is crucial to study these complex adaptive processes. So our model aims to shed the light on how the environmental factors trigger epigenetic changes by providing a computational framework to analyze these interactions and their impact on neurodegeneration. So the following study has been the results of this study by Song et al. From 2011 was used for this adaptive model network. So what it is, that the results of the study have shown that paraquat reduces the HDAC4 and 7 protein. The HDAC protein is a histone deacetylase, so it removes the acetyl group. Going back to that, paraquat reduces the HDAC4 and 7 protein expression levels, leading to increase of histone 3 acetylation. And this histone 3 hyperacetylation potentially plays a role in inducing dopaminergic cell apoptosis by activating mitochondria-dependent pathway, suggesting that this hyperacetylation may play a role in the pro-apoptotic pathway in the dopaminergic neuronal cells following a treatment with this neurotoxic paraquat pesticide. Um, and for modeling, we're using MATLAB. Yes, MATLAB 2024. So moving on to our um, model dynamic system. So first of all, it's a hierarchical control levels, and the vertical system biology was taken into account when modeling this adaptive network. The red arrows that you see here are hierarchical controls, while the blue arrows are adaptive modeling processes. So I'll start with you in the base level. So at the base level, we're modeling how into environmental exposures like using of the toxins interact with the dopaminergic neurons to influence Parkinson's disease symptoms. Specifically, in this level, this level captures the survival or death of the N27 mesencephalic dopaminergic cells, which is directly tied to the response to these stimuli. So in case there is no response to the stimuli, you have then we can say that the dopaminergic cells are dead, which is one of the characteristics of Parkinson's disease. Moving on to the first order. So next we have the first order adaptation level, which focuses on the brain structure and physiology. Here, the W state controls the strength of connections between environmental stimuli and neuronal responses. Whether these connections are preserved or lost depends on the health of the dopaminergic cells, which in turn is regulated by the enzymatic activities in the second order level. So before going into the second order level, I mentioned that before that the HDAC is acetyl deacetylase, is a histone deacetylase. So when going to the second level, we dive into the production of enzyme. This includes the HDACs, as you can see here, which negatively impact the cell survival. And these enzymes are also included in apoptotic cascades. So, and these processes here are influenced by the mRNA translation and can lead to dysfunction or cell death. So moving on to the mRNA production. Here we focus on the mRNA production, as I mentioned. This is where specific genes are transcribed to produce the proteins needed for enzymatic activity. For example, the mRNA linked to HDAC enzymes, which negatively impacts the cell survival. Um, and those are regulated by the fourth level order, which is genes. So this is at the fourth level, gene expression drives the mRNA production, as I said. This is where specific genes are transcribed to produce the proteins needed for enzymatic activities. For example, mRNA linked for HDAC enzymes, or mRNA that also affect the pro-apoptotic or the apoptotic proteins, which are amplified here, and those are reduced in this case. So moving on to the last, finally, the top level. Here we explore the gene expression of the epigenetic mechanisms. I'm sorry. These mechanisms controls expression of the key genes, such as I mentioned the HDAC and the histone acetylation, genes that are involved with the histone acetylation. So while some pathways like histone acetylation are understood, The exact epigenetic processes behind the HDAC regulations remain unknown. So for our simulation experiment, I'll go to the next slide for the simulation. So for our simulation experiments, we used MATLAB. The network characteristics are specified by role matrices, which define the parameters, which I'll talk about now, discussing the functions that we used for this model. So we used adaptation mechanisms. Two things we used to manipulate our MATLAB or our model was a combination. We used combination functions. We used the mathematical function like the logistic and the step ones. Those help to aggregate the impacts of incoming connections, adding flexibility to how the model processes the incoming data. We also used the speed factors. So, but by modifying these speed factors for each state, also combined by modifying the steepness and the excitability threshold, we controlled how fast and strong each state adapts to changes, which allowed us to have different rates of responses within our model. Let's see, and now I will show you our simulation. So the model captures adaptive changes at multiple levels, as I showed you before, including metabolic regulation, epigenetic control, and also gene expression, which illustrates how the cells respond in a dynamical way to environmental changes, like the paraquat exposure in our case. Before, as you can see here, this is before the paraquat exposure. The stimulation demonstrates a typical molecular processes under normal conditions. The key regulators like the HDAC is expressed and help maintain the cellular balance by de-acetylation. In this state, there is no detectable hallmark of Parkinson's disease, which were the not responding to stimuli, as I mentioned before, such as also which is linked to dopaminergic cell death. But when paraquat exposure begins, in this simulation is at alpha is equal 50, as you can see. This showed this significant disruption of the cellular processes. And this was obtained by manipulating the matrices of the MSFB, which stand for the combination functions parameters, the alpha and the beta. Yes. So after that, the simulation started again, but after the death of the dopaminergic cells. So let me repeat: from 50 to 10 is to 100 is the exposure, but then the exposure is stopped. But of course, the cells will not the cases will not return to the normal before the 50. So there is cell death that happened. So after the death of the dopaminergic cells, the W state, which I can show you, I will go back to make it more clear. So the W state here negatively affects the metabolic regulation. Going back to the simulation. And this consequently due to the cell death because it does not increase after the paraquat exposure. So the HDAC levels do not revert to normal values. Additionally, the states associated with external and internal stimuli as well decline until they are not detectable, even after removing the paraquat exposure, which indicates also cell death. But however, with the time, the histone 3 acetylation was also dramatically reduced, as you can see here. And this aligns with the results of the study that we used to model this network. Um, yes. I think I can go to the next slide. Um, yes. So now that we have seen the results, let's discuss the significance of this model, its limitation, where we can go from here. So regarding the significance, to start, this is the first computational model to link epigenetic changes with environmental and cellular interactions in neurodegenerative diseases like Parkinson's disease, as we used in this model. We think this is a big step forward because it helps us understand how these interactions play out across multiple biological levels. For the more for the relevance. This brings us also to why this model matters. It provides a foundation for developing more advanced tools, such as those for evaluating maybe therapeutic options and predicting potential side effects. It is also an important step towards better preventative strategies for Parkinson's disease using this model. Uh, Moving on to our limitations, because of course there's no model without limitations. So our model doesn't capture all pathological features linked to paraquat exposure, as highlighted by recent studies. And additionally, this model still requires empirical validation, so we can be able to refine its parameters and improve the prediction accuracy. And last, so what's next? For future work, we need to focus on several areas. So we need more research into the upstream processes that contribute to the dopaminergic cell degeneration. We need more in vitro and in vivo studies that are essential to establish that. Additionally, studying the duration and the concentration of paraquat exposure is also crucial for us to be able to refine our parameters. And finally, and most importantly, aligning this experiment data to refine our prediction parameters of this model. And by that, the presentation ends. And if you have any questions, we'll be happy to answer them.